Good afternoon, everyone. Today, I would like to present my research entitled Isolation and Characterization of Gibberellin-Producing Drought-Tolerant Bacteria from Chuping Perlis, Malaysia. Most existing studies on drought-tolerant bacteria have been conducted in regions that are well known for severe drought, such as West Africa and Egypt. However, Malaysia is also increasingly affected by the climate variability, including unpredictable droughts and floods. These environmental changes pose serious challenges to agricultural productivity. One notable area is Chuping Perlis, which is recognized as one of the driest regions in Malaysia and has been recorded temperature as high as 40.1 degrees Celsius. Despite this, Chuping remains an important agricultural area, making it an ideal location for studying drought-tolerant plant-associated bacteria. Plant growth promoting bacteria, or PGPB, are beneficial microorganisms that enhance plant growth, especially under stress conditions. These bacteria can improve nutrient uptake and produce phytohormones that support plant development. One important phytohormone is gibberellin, which play a major role in seed germination, stem elongation, and flowering. This study aims to answer three main research questions. First, how many bacteria can be isolated on tryptic soy agar from soil in the Chuping district Perlis? Next, what characteristics of the isolated bacteria can produce gibberellin from soil in the Chuping district Perlis? And lastly, what species of isolated bacteria can produce gibberellin from soil in the Chuping district Perlis? Based on this question, the objective of this study are to document the number of bacteria isolated on TSA from soil in the Chuping district Perlis. Next, to characterize isolated bacteria that can produce gibberellin from soil in the Chuping district Perlis. And lastly, to identify species of isolated bacteria that can produce gibberellin from soil in the Chuping district Perlis. By identifying and characterizing drought-tolerant PGPB, this study contributes to the development of environmentally friendly strategies to improve crop productivity under drought stress. In the long term, the use of beneficial bacteria may reduce dependence on the chemical fertilizer and help mitigate environmental pollution. Next, for the literature review, Chuping was selected as the study area because it is not only one of the driest locations in Malaysia, but also well known for its agricultural activities, including the cultivation of harum manis mango and MD2 pineapple. Farmers in this region frequently face challenges related to the drought stress, which affects crop yield and sustainability. Therefore, studying soil bacteria from this environment provides valuable insight into the microbial adaptation to drought conditions. PGPB are beneficial bacteria commonly found on plant roots or in the surrounding soils. These bacteria promote plants' growth through the several mechanisms, including enhancing nutrient uptake and producing phytohormones. Previous research has demonstrated that certain bacterial species such as Bacillus albus LRS2 are able to help crops like little millet adapt to the drought stress. However, it is also reported that different bacterial species may have preference for specific crops, indicating the importance of studying local bacterial population. Phytohormones are plant signaling molecules that play essential roles in the controlling plant growth, development, and responses to the environmental stress. There are several phytohormones discussed in this study like auxin, abscisic acid, and gibberellin. This hormone regulates various physiological processes and helps plants survive under the stressful conditions, such as drought. Studies have also shown that gibberellin interacts with the abscisic acid through hormonal crosstalk to balance plant growth and adaptation to the environmental stress. This interaction is crucial for plants to maintain growth while responding to the drought condition, making GA an important focus in this research. To identify drought-tolerant bacteria, several screening methods have been reported in previous studies. The PEG assay is the most commonly used method as it stimulates drought stress by reducing water availability in the growth medium. Other screening methods include the sorbitol assay and seedling assay, which are also used to evaluate bacterial performance under drought conditions. In addition to drought-tolerant screening, bacteria also can be screened for their ability to produce gibberellin. This process involves culturing bacteria in the nutrient broth supplemented with the L-tryptophan, followed by the extraction and the quantification of the gibberellin. For the methodology, soil samples will be collected from Chuping Ville, Jalan Haji Ahmad, and stored at low temperature before analysis. The samples will undergo serial dilution using the phosphate buffered saline, and soil bacteria will be isolated on a nutrient agar and tryptic soy agar plates. After incubation, distinct bacterial colonies will be selected and preserved for the future study. To screen for drought tolerance, the bacterial isolates will be grown under the stimulated drought condition using the polyethylene glycol or PG6000, and then bacterial growth will be measured using a spectrophotometer, and isolate that show good growth under the drought stress will be selected for the further analysis. These selected bacteria will then be screened for their ability to produce gibberellin by culturing them in the nutrient broth supplemented with the L-tryptophan. The gibberellin content will be quantified using the spectrophotometric methods. Further characterization of the bacteria will include gram staining and basic biochemical tests such as catalase and oxidase assay. Finally, molecular identification will be carried out by extracting bacterial DNA, amplifying the 16S rDNA gene using the PCR, and analyzing the result to identify the bacterial species. This is the flowchart for the method and shows the objective that will be achieved in the step of the isolation of the soil microbes, biochemical characterization, and the molecular identification. From this study, several outcomes are expected. Drought-tolerant bacteria that are able to produce gibberellin are expected to be successfully isolated from the soil samples collected in the Chuping Perlis. These bacteria will be characterized and identified, and their plant growth promoting traits will be evaluated. The finding may also help to identify potential bacteria that can be used as a biofertilizer to support sustainable agriculture and improve plant growth under the drought condition. However, there are some limitations and laboratory risks in this study. Some plant growth promoting bacteria may require specific growth media, which could limit the number of bacteria that can be isolated. In addition, contamination may occur during bacterial isolation, culturing, or molecular analysis. To reduce this problem, different types of agar media will be used and proper aseptic techniques will be applied throughout the experiments. These steps will help minimize contamination and improve the reliability of the research. This is a Gantt chart for this semester and next semester planning. And these are the references. Thank you.